Mysore University. She pursued her MSc in biochemistry from Yogi Vemma University, Kadapa, Andhra Pradesh. She has been awarded leadership from and lecturership from CSIR, NET, and a Bronze Award for Best Presentation in International E-Conference on Biomedicine. She is currently a life member of Asian PGPR Society, Auburn University, USA, and a member of Association of Food Scientists and Technologists, AFST, India. She has a myriad of publications, book chapters, and a patent filed in her name. Being a head of several faculty development programs and workshops has facilitated Indian and international conferences, has helped her all her way through career in life sciences. So, welcome you to the Xylem second session of Xylem series, ma'am. And I think we are good to go.
Okay. Thank you, Dhanushri, for such a nice introduction. I think I should thank the entire team as well as Senthil sir who provided this opportunity to interact with the new students. I think few of my students are also joined here. So it's pleasure to give a lecture like this for the students. Okay. So with no waste of time, let's move into our today's topic. So my topic already you're familiar with that. So emergence and advancements of monoclonal antibodies. So in this session, I'm going to explain about how the monoclonal antibodies have been developed and produced, how we can synthesize them, and in what way the research is being progressing so that the monoclonal antibodies will be used in the therapeutics, means in almost all diseases. Okay, that's and all I'm going to cover over here. I think I'm audible, right? Is it clear?
Yes, ma'am.
Yes,
Yes, ma'am.
okay.
Yes, ma'am.
So before actually entering into the monoclonal antibody production, so just have a brief intro about the immune system and what are all involved in the immune system. So the immune system, you all know, and almost all are the UG student and few PG student, right? You may be knowing the introduction because about the immune system and all. Anyway, I'll be giving a brief intro. So some few 3 or 4 slides will be a brief intro of our immune system. So the immunity or the resistance offered by an individual to a particular antigen or a foreign particle is nothing but the immunity. Okay. So whatever the components that are involved in such immunity development, that entire system will call it as an immune system, right? In the present slide, you can able to see there are different cells and organs that are involved in the immune system, right? So here mostly there are 2 different lineages. One is the lymphoid lineage and the other one is the myeloid lineage, right? So in from these 2 lineages, there are different cells and organs that offers the resistance in the host system. Okay, so you can able to see several different types of cells as well as organs are involved in generating such immune system. See, mucous membrane, lymphatic vessels, thymus glands, bone marrow, spleen. So all these are the main components and we can say cells and organs of immune system. Okay, so when we are talking about the immune system and the immunity,
Excuse me, ma'am.
yeah.
Yes, sorry to interrupt. Is your slides moving? Like, are you changing the slides, ma'am?
Yeah, now it's been changed.
No, ma'am. Only the first slide is being shown.
First slide. Actually, in mine it has been third slide. Okay, shall I share again? Wait, because in my system it is moving now.
Yes, ma'am. Please do.
Now, which slide you are visible? Difference between is that one?
No, ma'am. Only the first one, ma'am. The
first one.
your
Okay,
name.
I'll stop sharing and then I'll start again. Okay.
Yes, ma'am.
So now it is in first slide, right?
Yes, ma'am.
Yes, ma'am.
Okay. Now?
Yes, ma'am. Now it's changed.
Okay, ma'am. Yes, ma'am.
If I keep in full mode, maybe it is not visible. Can I do like this? Is it okay?
Yes, ma'am. No problem.
Okay. So I think whatever just now I have explained is about the general cells and organs that are involved in our immune system. So in our body, we have different types of cells and organs that are actually showing the immune system. Showing the immunity, right? So we have lymphocytes as well as and other blood cells. They are all showing the immunity. Okay. It's more. Is the slide is moving?
Yes, ma'am.
Okay, so there are two different types of immunities, innate as well as the adaptive immunity, right? So whatever the immunity that has been generated by birth in an individual, that kind of immunity, we can call it as innate immunity. So after the birth, because of encountering with any antigen or any foreign particle, whatever the system immunity develops, that immunity we can call it as an adaptive immunity. So with respect to innate immunity, there are several cells that are involved in that, like blood cells. Here in this slide, you can able to see different types of monocytes, eosinophils. So all these are cells are offering from birth onwards the resistance. So such a kind of immunity, we can say it's a innate. So now, there are further different types of innate immunity based on the some of the barriers, like physiological barriers, chemical barriers. So here the point is that the innate immunity may be differed with respect to species or from place to place. That's what we are I have explained in the slide, like barriers. So based on the barriers, the immunity offered by one individual will be differed from the other individual. Okay, so now coming to the adaptive immunity. So whatever the immunity that has been generated after encountering with a particular disease or any antigen, or in simple we can say foreign particle, so that immunity we are calling it as an adaptive immunity, and it is again of two types, like active and passive. So here why we are saying it's an active or a passive means, because the system is actively involved in the production of antibodies. So based on that production, we can say that there is active immunity and then passive immunity. So when we are talking about an active immunity, the system or the host system will be actively involved in generating the antibodies. Okay, so here the generation of antibodies may be of natural or it may be through vaccination. So natural active immunity refers to that whenever we have been exposed to any foreign particle, any bacteria or any virus, or when we are exposed or when we got any fever, like, so during that time, our host system can able to recognize that foreign particle and it has to show some response. So that response is nothing but the production of antibodies. So naturally, after recognizing a foreign particle, our body is involved in the production of antibodies. So the antibodies are being producing from the B cells. Okay, such development and the active involvement of host system in the production of antibodies, we can call it as an active immunity. So in other way, we are making our body active by giving the vaccines also. So what is the principle behind the vaccine? Vaccine means here we are giving a attenuated strain, right? So when we are introducing an attenuated strain, we are making our body to be prepared so that when the same live strain is being entered, our memory cells could be able to recognize and then they will able to produce the antibodies. Okay, so this is what the active adaptive immunity. Next comes the passive adaptive immunity. So passive means the body is not actively involved in the production of antibodies. Instead, here we are generating the ready antibodies to the body so that the ready available antibodies can able to fight against that particular foreign particle. So this is the major difference between the active as well as passive immunity. Now we are coming to our topic now, because here we are saying that it's a passive immunity, right? Here we are introducing the antibody directly into the host system. So how we are introducing the antibodies? So this is where that comes the picture in the generation of monoclonal antibodies. So because of that, I have given just 2 to 3 slides introduction. Okay, so here in passive immunity, again, it is of two types. All of you are familiar with the maternal immunity, right? So the fetus is getting the antibodies from the mother, right? Either through the way of placenta or it may be through the breastfeeding. So here what is happening, or the mechanism is that directly the fetus is getting the ready antibodies from the mother. So it's a kind of passive immunity, and we can say it's a natural passive immunity. And the other way how we can able to generate the antibodies means through artificial mode. So directly we can inject the antibody into the host system. Okay. Now, when we are talking about the immunity, immune system and all, we will be using the most familiar words like antigen as well as antibody, right? So what do you mean by an antigen? So antigen is nothing but any foreign particle, means any particle that is not related to a host system. If at all it has been entered into the body, then we can call that particular thing as an antigen. Okay, so this antigen has one important property. So what is that property means? It can able to generate the immune response, or it can immunize the host cell. Okay, so here one point you should remember, that antigen will be a whole structure, right? So in that whole structure, the entire antigen may not able to cause the immunogenicity in the host system. Only few parts are the important parts of the antigen will be able to generate the immunization in the host system. So that particular parts, we can call them as epitopes. So within the antigen, the epitopes are present, and these epitopes are the actual immunogens which can able to cause the immune response. Okay, so in recognition of an antigen, our host system will develop a antibody. So this is how the antibody looks like. It's a Y shape. Okay, then here we can able to see there will be heavy chain and then line chain, and in general constant regions and then variable regions, right? So usually the entire antibody is also not involved in interaction with the antigen. Within the variable region, there are some constitutive regions which we are calling them as hypervariable regions. So that particular regions are actually involved in binding to an antigen. So that particular region is nothing but a paratope. So paratope of antibody is involved in binding to an epitope of antigen. That's why we can say in general, the variable region can be called as antigen binding site. So, and you all know about different types of antibodies are there. So the difference in the antibodies is because of the heavy chain difference, because each type will have different type of heavy chain. It may be gamma type, mu type, epsilon type. So based on that, the antibodies are further divided into 5 different types. Next. So now we'll see what is a polyclonal antibody. So now you all know that antibodies are generated in response to a particular antigen, or we can say in response to a particular entry of a foreign molecule. Okay. So whenever the foreign particle has been entered into the host system is immunized. So the main cells are the organs involved in that. In the antibody production is B lymphocytes. So from the B lymphocytes, the plasma cells are the ones which can able to generate the antibodies. Okay, so with recognizing a foreign antigen or a foreign substance, that B cells will be activated and they will produce different types of antibodies. So several B cells can able to produce different types of antibodies, and that antibodies can able to bind different epitopes on the same antigen. So here you can see in this diagram, this is an antigen with 3 different epitopes, epitope A, B, and C. So here a group of antibodies, and this particular antibodies we are calling them as polyclonal antibodies. Okay, so this polyclonal antibodies can able, means the hypervariable regions are the variable regions of these antibodies may be binding to any of this epitope. You can see here, you can observe. So the antibody can able to bind with the epitope A and with epitope C as well as with epitope B. Means there is a lack of specificity. So a group of antibodies that has been generated from a group of B cells, which can able to bind any epitopic region of an antigen is called polyclonal antibody. So this is also offering resistance, but this is not considering into importance. Why? Because here the most important thing is they are lacking the specificity. Okay. So when there is a lacking of specificity, then obviously the results will be very less. So if you are targeting to a particular disease and particular structure, then the polyclonal antibodies may not work properly, right? So that's why the research has further progressed in order to develop the monoclonal antibodies. So here polyclonal, how we can generate the polyclonal antibodies means just we can immunize the mouse repeatedly. We can immunize the mouse and we can collect the serum. So from the serum we will collect the active B cells and then we can screen, then we will purify the antibodies and then we can use them as a polyclonal antibodies. So this have specificity towards any type of means not to a particular target, but it show action on any type of epitope, okay, in this particular antigen which we are using as an immunogen. So this is of not much importance in the therapeutics. So we can say that research is further progressed in order to develop the monoclonal antibody production. So here, what is the monoclonal antibody? Means the group of antibodies that are being developed from a single B cell and which show specificity towards one particular structure. We can call them as a monoclonal antibody. Here the same antigen you can see, but the produced antibodies, they are all interacting with epitope B only. See here, you can make out the difference. So such a kind of antibodies which are being produced from a single B clone are called as monoclonal antibodies. Now, how we can generate this monoclonal antibodies? Okay, first, here we need two different types of cells in order to produce the monoclonal antibodies. One is the spleen cells and the other one is the myeloma cells. So spleen cells will be collected from an immunized mouse. So how we are immunizing a mouse means we are injecting the antigen repeatedly into a mouse. After a certain period of time, then we will collect the spleen cells from the mouse. Okay, then we are mixing the spleen cells with the myeloma cells also here. So why we should need myeloma cells? Because spleen cells has certain lifespan. After that lifespan, they are going to die. So during that period, if we are using only the activated spleen cells, then they may not be able to give in a long run. Means in a long run production of antibodies, because of shorter lifespan, they will be dying. So our antibody production will be inhibited or stopped in between, right? So in order to have a longer run of antibody production, now we are using the myeloma cells also here. So myeloma cells are nothing but the tumor cells, all of you know. So main characteristic feature of a myeloma cell is immortality. That's why we are seeing in cancers, it's a continuous uncontrolled growth, right? So they don't have the death. So we can say that immortal myeloma cells can be used. Now we are fusing the spleen cells. So here spleen cells means immunized spleen cells, okay? Immunized with the desired antigen. Okay, that spleen cells are being collected from the mouse and then we are then mixing with the myeloma cells. We are allowing them to fuse in a polyethylene glycol medium so that they will produce a hybridoma cells. So this hybridoma cells will be screened and then further it is being analyzed for several steps so that we can finally get the desired antibody production. So this is in simple, we can say that monoclonal antibody production, but as I said, this is not that much easy to use because here the point comes. So how we can select the exact hybridoma cell which is made up of spleen and myeloma cell, is it not? Because there may be a chance of formation of hybrid from spleen and spleen cell. See here you can see, then the myeloma and myeloma cells also can be fused and they form a hybrid. Along with that, we will also get the desired spleen as well as myeloma cell. but we need to select from that medium where the hybrid is containing only the spleen as well as the myeloma cell. So for that, we need to provide a mechanism, right? So in order to select a hybrid cell which is of only spleen and myeloma cell, we need to provide one mechanism. So what is that mechanism means? Here we are relating that to the nucleic acid synthesis. So in general, we have two different types of nucleic acid synthesis metabolisms, right? One is the de novo pathway and the other one is the salvage pathway. So what is the difference between the de novo and the salvage pathway means? So in de novo pathway, from the base, from small structures, the nucleic acids will be synthesized, like they will organize it from the ribose and then they will synthesize the bases, then joining, like that. From the core, the synthesis will be starting in case of de novo pathway. Okay, so the other pathway is a salvage pathway. Here it won't start from the core. Instead, if any intermediary products are available, then this enzyme, the main enzyme involved in the salvage pathway is HGPRT, hypoxanthine phosphoribosine transferase enzyme. So this enzyme has the ability to convert the components into respective bases. See here one example, I had shown that hypoxanthine to guanine. So from the intermediates, it can able to generate the bases and then they are involved in the nucleic acid synthesis. So this is the salvage pathway. So here what we are doing is that we are blocking the de novo pathway by aminopterin. So the hybrid cells are allowed to grow in hat medium. Okay, in the hat medium, we are using the aminopterin. A refers for aminopterin. So when we are using the aminopterin, it will blocks the de novo pathway. Okay, so when the de novo pathway is being blocked, the nucleotide synthesis from the core will be inhibited. Okay, then the cells has to depend on the salvage pathway. So already I explained that in the salvage pathway, it needs some immediate precursor. So here the point is that you can able to remember that there may be a chances of three different types of hybrid cells, right? Spleen cell, that means B cell, whereas hybrid of myeloma and B cell, and then myeloma hybrid. Okay, so here one more characteristic feature of a myeloma cell is they are HGPRT negative. They don't have the HGPRT enzyme. So obviously in the myeloma cells, when the de novo synthesis is blocked, because as they are HGPRT negative, so they won't able to survive because there will be a no nucleic acid synthesis through the salvage pathway also. Then the complete inhibition of nucleic acids will be seen in the myeloma cells. Then they may not able to survive. Okay, so though they have uncontrolled growth, but as nucleic acid itself is not there, then there is no survival rate. Okay, in a similar way, let's come to the B cell hybrid. So in the B cell hybrid, these are HGPRT positive and they can able to show the salvage pathway. But here, these are also cannot able to grow. Grow means in the sense they can able to grow, but they have a limited lifespan. So because of that lifespan, they will survive up to their lifespan. After that, they will die. Okay, so in that way, this B cell HGPRT cells are also being died. So now in the hat medium, we are left over with a hybrid cell that is showing one B cell and myeloma cell. So here the myeloma cell is HGPRT minus, but as it is a hybrid of B cell, so it will get the HGPRT from this and then it will able to survive. Okay, so only B cell will have limited lifespan, but now it is a hybrid of myeloma cell, so it will acquire the characteristic feature of immortal growth. So in this way, we can able to select the hybrid cells from a hat medium. So here it is clear that whenever we are saying that is a spleen and spleen cell hybrid, they may be die after their lifespan has been ended. Okay, then myeloma cells can't able to grow in the hat medium because aminopterin blocks the de novo pathway, and these already know that they lack the HGPRT enzyme, so they cannot able to go for salvage pathway. So these cells also died. Now the hat medium is left over with only hybridoma cells where we have our desired cells. So from this we can select, isolate these hybrid cells and it is not easy also this step, because there will be a chances of more and more contamination. So these are all the steps we need to consider when we are preparing the hybridoma cells. So the research is progressing, because research means it's not developed in one day, right? So many steps we need to do, and so many things we need to face, then only we can understand, or we will guess, we will hypothesize. So which way we can proceed and what. So though we are getting the hybrid cells and we can able to select only hybrid cells, but there will be a chances of contamination. That's why I'm saying the monoclonal antibody production is not that easy what we are seeing. So without contamination, we should isolate, then we should take further. Here there may be a different types of clones, and then serial dilution we need to do, so that because of this serial dilution, finally we will get a single clone of B cell where it can able to generate the antibodies which are of monoclonal type. Means what we have immunized to mouse, that we can able to see here. See, this is what the entire monoclonal antibody production. I think it's clear. Did anybody has doubt in this? Is it clear?
Yes, ma'am. So I just have a small doubt. Like, is it that the salvage pathway that is being occurred in all the cells, or is it de novo pathway too?
Actually, in both the pathways have been occurring in a cell. Okay, whenever one is blocked, then it has to survive, during that time. So salvage pathway means already intermediates are available. From the intermediates, we can able to see the nucleic acid synthesis, but mostly the de novo pathway, because from the core, it can able to synthesize, no? So in the cells, both the pathways we can see.
Okay. Okay. Thank you.
Is it clear? Okay, shall I move further? Means the selection.
Yes, ma'am. You
It's
can
important.
continue, ma'am.
Hat selection. That is the main thing for the monoclonal antibody production. Okay. Okay. See here, now when we are getting the monoclonal antibodies here, how they will work means, because they have specificity towards a particular epitope. So this is the antigen, then all will show, will act to this antigen at this particular epitope. So then all can go and bind, and they will make it harmless. See here. That means they will make, they will suppress this particular antigen, and then they will do its action. That means this monoclonal antibodies can able to eliminate the entered foreign particle over here. So here this may be linked to any sort of diseases. So okay, after seeing all the monoclonal and polyclonal antibodies, now the major differences we can find out. This is monoclonal, as I said, it is coming from a homogeneous or single B clone, whereas the polyclonal, it refers to the heterogeneous B cell production, and then antibodies are coming from different B cells. So because of that, we may say that the cross reactivity is very high with respect to the polyclonal antibodies, whereas with respect to monoclonal, the cross reactivity is also very low. Okay, and one more thing is that because as we discussed just now, the monoclonal antibody production is not that easy. In each and every step, we need to take very care contamination, and also it is expensive, right? Because we are specifically targeting one particular site, so it is expensive when compared with the polyclonal antibodies. So these are the major differences you may find between the monoclonal and polyclonal. But on whole, we can say that the monoclonal antibody will work efficiently when compared with the polyclonal antibodies. So the research has not started there itself. Okay, up to now we had seen that in the passive immunity, directly when we are injecting the antibodies, they will show the action towards a particular disease, right? So here, for that sake, from the polyclonal, we are moved to the production of monoclonal antibodies. So again, the research further progressed, and we can able to see different types of monoclonal antibodies here. Like you can able to see the murine, chimeric, humanized, and then other chronic therapy used antibodies. So murine means it is nothing but completely it has been generated from a mouse cell. So in the synthesis we had seen, we are immunizing a mouse with the desired antigen, right? So to which we need the antibodies. Like, so there in the murine antibody, whatever the antibodies here when I'm talking about the antibody, it's all about the monoclonal antibodies. Okay, so the antibodies that has been generated from this murine is completely of mouse based. Okay, so that's why we are saying it's a murine antibodies, and this type of antibodies may sometimes show some allergic reactions because it is coming from a mouse source, right? So this has some limitations. It has positive and then it has some limitations. So in a similar way, further research has progressed towards the production of chimeric antibodies. So what are the chimeric antibodies means? Here there are two fusion of two different antibodies. Okay, so we can say that the whole thing up to the variable region, whatever the antibody we can able to see, that is from human. So here you can see. So then we are fusing this human antibody with the variable regions of that are coming from a mouse. That's why we can see these are all the parts, variable regions that had been acquired from the mouse, and then rest of the thing is from the human antibody. So in that way, the chimeric antibody will be generated. And here we can say that 65% of human antibody. So there will be a less chance the allergic and other non-specific reactions. Okay, this are also can be used. Then further research has progressed towards the humanized monoclonal antibodies. Here, what is the difference between the chimeric and the humanized monoclonal antibodies is, in both the cases, the main thing will be from the human, like in the Y shape, whatever the constant region and variable region is there. In case of humanized antibody, it is from the human. But as I said in the first slide, when I'm explaining about the paratope, so within the Y shape or within the variable region, there are some hypervariable regions, or we can say the paratopes. So here only paratopes are being taken from the mouse and then they are fused with the human. So we can say that completely 95% they are of human type. So there will be a low not immunogenicity, low allergic reactions with respect to the human individual, because they're completely of human type. Only the hypervariable regions are being collected from the mouse and they had been introduced into the sim. So with respect to the binding capacity, because we are directly adding the hypervariable regions here, so there may be weak interactions with the antibody also. And the research has not stopped there itself. It has further moved to produce a complete human monoclonal antibody. So what do you mean by that? By this human complete monoclonal antibody is nothing but, so we have been collecting a immunoglobulin gene. So immunoglobulin gene can able to produce the desired antibody. So the immunoglobulin gene from the human we can able to collect, and then we are introducing or fusing with in a mice, and then we are immunizing the mice with the respective antigen. Then the immunoglobulin gene, upon recognizing that foreign particle or the antigen, it can able to undergo the expression. So that expression is nothing but the production of antibodies. So here whatever the antibodies are being produced, they're completely from human source, because the genes are collecting here from the human and that genes are fusing in the mouse. Then we are immunizing the mouse so that the genes, human genes are activated there, and then they are expressed, and then whenever there is an expression, then obviously that leads to the formation of different antibodies, right? So that antibodies are again using for human purpose. That's why we are saying it's a human monoclonal antibody. So here all the things are simple. There is a minor difference. See, murine antibody is completely from the mouse based, whereas with respect to the chimeric, so the up to the variable regions it is from human origin, and the from the mouse the variable regions are collected and they are fused with the human immunoglobulin. Then we can say that kind of monoclonal antibody as a chimeric. And then we are calling one more as a humanized. So here the entire thing is from human only, immunoglobulin, only a part, that means hypervariable regions. So the amino acids that are related to the hypervariable regions are being collected from the mouse and they are being introduced into the human immunoglobulin. Okay, then we can say it's a humanized almost 95%. And one more, through the genes also we can See, so here with respect to genes means the immunoglobulin genes from a human are being introduced into a mouse, and then we are allowing to express in the mouse, then we'll collect the antibodies. So they are nothing but recombinant, we can say. So their complete humanized antibodies are also being seen. So these are all the different types of monoclonal antibodies through which the research has been progressed. So here you can see the examples for all the types, and these are all see the names now you are familiar, right? Mouse, chimeric, humanized. So when you see this, you'll know that mouse antibody means it is completely from the mouse only. Chimeric means okay, human antibody is there, along with that mouse variable regions are there. Okay, so and whatever the down things you are seeing, these are all FDA approved monoclonal antibodies, okay, of each type. So these are all the names. We can say that they are chimeric monoclonal antibodies, and then they are approved by the FDA. So that now they are using in the therapeutic uses. So all these are examples for different types of monobodies. I mean monoclonal antibodies. See, mouse. So all now we are using all the different types. Means for a particular disease, we may generate chimeric. Means based on the disease type, we may see the mouse or chimeric or humanized, anything over here. Okay. So this is all about the monoclonal antibodies and how the research has progressed and what are the genetic engineering variations, everything. Now we'll move to the final session, like applications of monoclonal antibodies here. So now we had seen different types of monoclonal antibodies from basic to today. The monoclonal antibodies are being using in different fields. See, in the therapeutics itself, in almost all areas, the monoclonal antibodies are being used, like in the diagnostic biochemical analysis and some cancer treatments, transplant rejections, infectious diseases, or for your laboratory experiments, everything, and some protein purification mechanisms. So in all those things, we are using the monoclonal antibodies. Okay, so because of time constraint, each and particular disease I am not going to explain here. One slide I will explain, because the monoclonal antibodies will be generated to any SAR, and you can clearly observe this. Maybe I'll read out the antibody, brand name, okay, it's FDA approval date, and type, target, and indication. Okay, so here you can able to see different types of monoclonal antibodies. They are targeted to different diseases. See here, cardiovascular, autoimmune diseases, transplant rejections, and some other. Still it is continuing. See here, multiple sclerosis, and within the cancer also there are different types of. So for all these types of diseases, there are available monoclonal antibodies. So these are all the names that has been given to the monoclonal antibody generated to against disease, and as one pharma company has been developed that one, based on that the brand name is also being different. And you can see the important thing is the type. So just now I explained about different types of monoclonal antibodies, right? See here, for example, for the cardiovascular disease, chimeric type of monoclonal antibody has been used. That means you got an idea that chimeric means, okay, here almost up to a constant region, it is from the human origin. That means the mono antibody will be, and only the variable region is taken from the mouse. So like that, you can see the type. So almost all the types are being using for different diseases. Is it clear, students? This one?
Yes, ma'am.
Yes. Okay. So
Yes,
by
ma'am.
seeing it, you can get the idea. Okay. Now you are you will understand that okay, this is a chimeric type of monoclonal antibody. So most of the thing is from human, like that. So here you can see for almost all diseases we have the monoclonal antibodies and some specific. So within the inflammation, you can able to see different types of monoclonal antibodies, and we have different types of targets. So here also you can see one more region called target. So where it is actually going and binding, and where it is inhibiting, or where it is showing the action towards the foreign particle. This is the target region. You can able to block this. So as a result of that, the foreign particle is going to suppress. So here the foreign particle may be bacteria, virus, virus or any toxic compound from the bacteria or virus or any other substance. Okay, so this whatever the developed antibody will go and bind to the toxic compound, and then it will able to destroy. So finally that leads to the disease. Okay, then we can use that in particular disease, so we can save from the people from those diseases. Okay. So within the inflammation as well as within the cancer, you may able to see different types of monoclonal antibody, and all these are, remember, these are all FDA approved. That means Food and Drug Administration approved and currently using also. See how the research has been evolved. So we have studied the monoclonal antibody production, but the research has progressed a lot, and now we can able to see these many antibodies. See, it's a great thing, right? So these many antibodies can able to cure different types of cancers, and they are used in different diseases. Okay. See, and along with the cancer, not only for cancer, almost all diseases. And one more simple things I can give the uses of monoclonal antibodies in your laboratory experiments. Also, you may be able to use the monoclonal antibodies. Like I think PG students, they might have been doing the radioimmunoassay, and then the pregnancy test. So in that we'll use whatever the antibodies, because they are specific to a particular antigen, right? In order to detect the levels of that or the presence of that component, we will use the monoclonal, because this antibody should be specific to that particular antigen, right? So in such simple experiments to massive diseases, the monoclonal antibodies have been using in our life. Okay, now this is all the overview. So to the present scenario, one, I want to explain in clear the usage of monoclonal antibodies now, because we are facing the pandemic situation due to COVID. So the monoclonal antibodies are also used for arresting the coronavirus. So just I'll explain this a bit more because all those applications are there, and presently up to COVID also we are using the monoclonal antibodies. So here you can see that how we are activating the immune system in the COVID situation or in any others, means through the vaccines, right? So whenever we are vaccinated, that vaccine can teach our body that a foreign particle has been entered, and we are giving a attenuated strain. Attenuation means weak strain, okay, which cannot able to cause the harm, but it can able to cause or generate the immunogenicity. Okay, so that vaccine can teach our immune system, okay, you are being encountered with a foreign particle, so the B cells of our immune system has to be active, so that they need to produce the antibodies. Like that our body will be prepared because of vaccination doses. Then what will happen when our B cells will have effector B cells and then memory B cells. Okay, so effector B cells, they will do the action immediately. Okay, whereas the memory B cells can keep it in mind, and whenever the live cell has been entered, live coronavirus has been entered, the memory B cells will play a role, major role, and they will recollect that, okay, already we are vaccinated, our body is ready with that, so it can immediately produce the antibodies and it can able to kill the virus. Okay, so this is the main mechanism behind the vaccination, and we can see. So here this is through the vaccination, but with respect to monoclonal antibodies also, in this we are directly providing the monoclonal antibodies to the human system, okay, or to the affected people or to a normal people. So here the monoclonal antibodies will be readily available, and these antibodies, how we are collecting means, so we need to collect the blood sample from a COVID recovered patients. So that is what we are doing, right? So we should take the blood from recently COVID recovered patient, and from that blood, what we need to do, we need to isolate the exact cells where exact clone of B cells where they have the ability to go and bind to the spike region of the COVID virus. Okay, so we need to identify and isolate that particular cells, and then we need to isolate the DNA also, means genetic information we need to collect, and then further we need to propagate so that bulk amount of monoclonal antibodies will be generated. So in this way we can able to generate the monoclonal antibodies against a spike protein of SARS-CoV-2 vaccine, and we will introduce into the human system. in order to provide the protection. But here we can say that there will be a benefits and there will be some limitations in collection of this kind of antibodies, right? So potential benefits means we can offer the prevention before a vaccination, because usually vaccination procedure, if it is a new, like in this pandemic for COVID, we had seen in order to develop a vaccine for any particular type of new disease or any infection or any bacterial virus, anything, it almost takes 2 to 3 years, because though we have been generated, several clinical trials has been done. Without that, FDA or anything cannot approve that vaccine, right? So even you might have been aware of that, even in our COVID situation also, the vaccine is released within a year, because of the situation that is really worse. It's this massive spreading and all. So because of that, the prior to vaccine, because in order to develop the vaccination, it is a time taking, but this kind of monoclonal antibodies can give immediate action, right? So until we will wait for the vaccination production, if the human system is being providing with the antibodies, then the antibodies can able to fight with the foreign substance, or we can say here the virus. But this is the potential benefit we can say, but there is a limitations also, because our immune cells is not immunized, because they are readily available with the antibodies. So it's the resistance offered by the immune system will be a short limited, because only if the given antibody number has been decreased or reduced, then because of no memory recognition, our immune cells cannot able to produce the antibodies again. So in everything, we can say there will be a benefits, means positive effects as well as the negative effects. So vaccination will take time, means vaccine production will take time, but there will be a memory response and it will be a lifelong run. Whereas immediate monoclonal antibody treatment will be, the treatment will be immediate, the effect will be immediate, but there is no memory response here. Okay, whenever the same person has been encountered with the virus again, then there will be no memory cells, right? Then immediately, okay, so he may not able to offer the resistance. That's why we can say that there will be a positive potential benefits as well as some limitations with respect to monoclonal antibodies. But anyhow, we can say that this monoclonal antibodies are being using in present COVID situation also. So here, this is the COVID-2 structure. Mostly the monoclonal antibodies are being producing to the spike. Okay, to this particular spike, the antibodies are being producing, and then these antibodies are going and binding to these spike regions. Okay, so whenever the SARS-CoV-2 virus is being bounded by this monoclonal antibodies, they will lose the capacity to enter into the human cell. That's why we can reduce the spreading of virus into all cells of our body. Okay, so this is all happening because the spreading is arrested because of the monoclonal antibodies and they are binding to the spike. Okay, then okay, these are all the monoclonal antibodies that has been generated for the COVID-2. I'll show the mechanism names. So these are all the names presently what we are using. See here the mechanism. So whenever the antibody is being bounded, it prevents the entry of virus into a normal cell. Instead, this monoclonal antibody on the one side, means the variable region, can able to recognize the spike protein, whereas on the other side, it can able to recognize the ACE2 receptors of endosomal cells. So whenever the ACE2 receptors recognizes this, the endosomes will engulf them, and then finally it leads to the destruction of virus. So here, because of this antibodies, what we are seeing, we are reducing the spread or entry into a normal human cell. Instead, they will allow the entry into a endosomal cell where the destruction of virus cell is being happening. So in this way, the monoclonal antibody production is being using in our present COVID pandemic also for the SARS-CoV-2 virus. So here one way we have ACE receptors which can able to recognize this monoclonal antibody at the constitution. Other way, they have some other receptors also in the constitution that also can able to recognize the endosomal receptor cells, and then they will be finally entering into the endosomes, and finally the viral genetic material will be inhibited or will be destructed. So in this way, the antibody antibodies, whenever we are introduced into the cell, they are going to kill the virus. So this is how we can able to prevent the COVID virus entry into all cells and finally the COVID disease. Okay, these are all about my today's topic. And okay, there may be side effects, right? So up to now we had seen the monoclonal antibodies are all being involved in curing several diseases, but with mild to severe side effects are also there. Okay, so whenever FDA releases one kind of monoclonal antibody, means some tolerable side effects will also be there, like some kind of rashes, diarrhea, nausea and all. So if they are also there, then the FDA will approve, because these are the minor things that can be treated, right? But if some monoclonal antibodies will show severe diseases like that leads to death, that won't be approved by the FDA. So we are seeing most of the positive scenes, and along with that some cons will be there, right? So but the things will be reduced, minimum things will be reduced, and there will be allowed. But if the things are too worse, then they're not at all allowed. Okay, so this is all about the monoclonal and the research, how it is progressing. So I'll conclude this session. See, now we had seen the monoclonal antibody production from past to present COVID situation also, right? So that means the technology has been developed so much and new therapies are being evolved. So because of this, we can able to cure from simple diseases to the complex diseases, right? But one factor should be, we should keep in mind that whatever the monoclonal antibodies will be generated, and the pharma companies are all interested, so they should be able to manage and treat all the diseases, all the conditions with respect to clinical and cost-effective process. So they should be keep in mind in order to generate any sort of monoclonal antibody. So that's all about my session. I thank all the organizers like Senthil sir and then Dhanushri for giving this opportunity. So now it's open for queries. I hope you understand from basic to advanced research. I think so. Yeah. Dhanushri, from your side.
Yes, ma'am. It was really a very insightful session. Like we go for a question and answer session now. Like we have another 8 minutes with us. So I request the audience to please post the questions in the chat box only, so we can take up the questions in a very organized manner. And yes, ma'am. So we can go with the first question.
Okay.
Okay, so the first question is from Dhanushi herself. Did both salvage and de novo pathway fail in the function of immunity of human body during the pandemic?
Can you repeat? It's not clear. Or can I see the chat?
Did both salvage and de novo pathway fail in the function of immunity of human body during the pandemic?
During the pandemic, why? Okay, in the pandemic situation, will the both pathways are blocked? That's what your question, or?
Yes, ma'am. Failing
I
the
think
function of immunity.
actually what is happening in this pandemic, because of virus entry, it is actually I mean, destructing the complete immune system, right? So when the immune system has been destructed, there is no option for the cell to survive. So during that time, even the synthesis are there or not, the cell cannot able to survive, right? Because of lack of complete immunity.
Okay, thank you, ma'am. The next question is from Abhilash Babu. What is the difference between monoclonal antibodies and vaccines?
Okay, right. So vaccines means we are giving an what we can say that attenuated strain into the host system. We are introducing a attenuation is weak strain, heat inactivated, or there are different process of attenuations. Means a organism which can be attenuated, like bacteria you take. So bacteria will be attenuated, and then we'll introduce that into the individual. So what is happening here? The individual will treat this as a foreign particle and start responding to it. And we are attenuating the toxicity. So it can able to generate the immunogenicity, but it cannot cause any harm to the host system. So the host system can able to recognize that as a foreign particle, and the host system can able to generate the antibodies by its own. So the B cells of the immune system are recognizing that structure and they will produce the antibodies here. Okay, this is all what the vaccines will do. So in the vaccination, we are introducing only a part of organism or attenuated organism so that the body will be immunized and body will start producing the antibodies. Whereas in case of monoclonal antibody introduction, directly we are providing the antibodies to a host system. Okay, so the host system through the injections, okay, then the host system will contain a ready-made available antibodies and they can fight with the disease. So this is what the difference, because here directly we are providing the antibodies, but here we are making the body to produce antibodies through vaccination.
Here I have to, excuse me, this is Dr. Sandil.
Yes, sir, hi.
Hello, how are you, Dr. Sonlada?
Yes, sir.
Yeah, actually that for previous question, sorry for the interference. See, both monoclonal as well as vaccine is a totally different mechanism. Okay,
Yes,
so one is we are going to artificially making that monoclonal antibody from the selective single origin of the cell or cell line. But this case vaccination, we are going to produce for the 4 different mechanisms. Say, for example, attenuated and live, and we can
different strains are there.
Different. Okay, so it's a kind of both vaccine as well as monoclonal antibodies. The different mechanism can obtain in the human organism. Okay, that's what. Are you okay, Abjit, or who is that? Hello. Hello.
I believe
Yes, sir,
got it?
Yeah, yes, sir.
Okay, okay, thank you. You are Abjad, right?
Abhilash.
Abhilash. Oh, Abhilash, you are got it now?
Yeah, yes, sir.
Okay, thank you. Thank you. So over to Dhanasri. Over to the Dhanasri. Thank you.
Okay, thank you for the answer, sir. So the next question is, can you differentiate between monoclonal antibodies and convolution plasma therapy methods in treatment of COVID?
Plasma therapy and then monoclonal.
Methods in treatment of COVID, yes, ma'am.
Yeah, in plasma therapy also, I think we are collecting the plasma, but again, we need to go for further selection of exact B clone, and then we need to identify the antibody or the gene that can able to produce the antibody, which can able to bind to the spike. Okay. So even though we are collecting the we are transferring the plasma cells, but further selection to be done for suppressing of that spike.
Okay, and then the last question is, how monoclonal antibodies are used in the treatment of HIV?
HIV. Actually, I'm not sure about this answer. Means whatever the basic knowledge I have, and means exact mechanism what are using that I don't know. But I will let you know to Dhanasri for this question. Means exact mechanism I don't know actually, because which receptor and all. Concept is same, but exact receptor and all I may not say now. Because particularly you are asking about the HIV, so I need to study which receptor and what is the mechanism behind that. Okay, I'll let you know that one.
Cool. We can revert back with answer later. Ma'am, also I have another question. Like a drug usually takes 50 to 20 years to come into market, right? So did they accept the COVID vaccine in less than 2 years? So
can you repeat, because your voice
how do
is breaking?
we trust the vaccine?
Can you please repeat the question, because there is a audio disturbance? I think Dhanasri, it's yourself.
Ma'am, her question is, a drug usually takes 15 to 20 years to come into market. So how did we accept COVID vaccine in like 2 years itself?
Yeah, that's what I explained. No, so there are some complications. So if it is not major complications, but the situation is here severe, right? because in the COVID we had seen it's a massive spreading. We cannot able to control, we cannot able to treat. We need more and more hospitals and all. That's why I'm saying a vaccine should be almost it should need 3 years, and then with lot of clinical trials, and then it should be studied from ages also, because when we are introducing a vaccine, how it is affecting from child age people to middle age to old age people, and with respect to especially the children. So what are the hormonal systems that may are going to affect with this vaccination process and all? They need to study, they need to do a lot of clinical trials, and then they will only release such a kind of vaccine. So if there is no any side effects, then only they will release. But here the COVID vaccine has evolved, though we can able, so out of 1,000, if one is getting affected, so that may be not counted by the government, because the situation is such worse. We had all faced that. That's why they have evolved. And that too, here also they have done the most important clinical trials, and then only they have released that. But to here also you should think that the young and the children, they are not yet released till now, because recently only about 12 we got, because they are in the developing stage. The hormones, everything are to be underdeveloped. So the external giving vaccines may affect the hormonal changes and all. So that may affect that particular children. So because of the massive destruction that is happening, they are going to they released that. So out of 1,000, if 1 or 2 is getting died or because of vaccine, that is not counted here, because in order to arrest the other worse situation, they released that one. But yeah, as you said, it need complete clinical trial. I hope I answered. Are you there?
Thank you, ma'am.
Yeah. Any other queries?
I think that is regarding the questions. Yeah. So if
Yes, ma'am. I think
we
her
will get
voice
back
is
to you.
breaking.
Share
She's
the,
facing
I share the.
some-
Is there any
Hello?
question? Hello?
No, ma'am, no. Ma'am. So her Dhanushi's voice is breaking up. She's facing an
Yeah,
internet
her
issues.
voice is not clear. It's
Yes, ma'am.
yeah.
Anyways, ma'am, thank you for the session. Today we were on time and you spoke really in-depth stuff, so it was really helpful. Well, I think we can share your email ID later on to others so that they can post questions.
Okay.
And thank you everyone for joining. Please do fill out the Xylem form that I have posted here so that you can join the Nakshatra Discord server and other platforms where you'll be able to know about upcoming series from Nakshatra itself. And yes, thank you all for joining. I hope everyone here enjoyed and learned new concepts. And yes, I believe that's all.
Am I audible? I just want to add
Yes,
something.
yes.
Yeah, hello, ma'am. I just wanted to thank you from Nakshatra's side. I also want to maybe suggest that if you have any particular questions, 1 minute.
Your voice is not audible, Dhanushri, I think. Or...
No, Sitara was
No,
speaking.
ma'am, it's Sitara.
Yeah. Hello.
Hello.
Ma'am, so I just wanted to say if you can suggest some, simple activities or questions that the students can go back and research a little bit more on, that would be really helpful, because we give 1 week for them to kind of focus on one particular topic. Like last week was a little bit more about oncology and cancer. So similarly, this week, the coming week, they have time till next Sunday. So if you can suggest 1 or 2 topics that they can go back, research a little bit and write a small short essay or report, that would be really helpful for the students.
Okay, I'll give to Dhanushri maybe by evening. Then you can share, I think, Dhanushri, to all your group.
Yes, ma'am.
Yes, ma'am. Please do that. That would be very helpful. So once again, thank you for your time. It is really an honor to have you on our platform. Thank you so much.
I wish to thank Senthil sir and this Dhanushri, all the organizing team, for inviting me for this talk. It's really wonderful to teach new students. Yeah, it's good. Okay, that's it.
Thank you, ma'am. Yeah, that's it. That's it. Have a nice day.
Thank you, sir.
Yeah.
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 Thank you, Senthil sir, again. And Swarnalata ma'am too. Thank you for being there with us today, ma'am.
Oh, thank you. Thank you all. Okay, I'll
Then
yes, sir. Senthil sir is going.
no, it's okay. It's okay. You are welcome, Dhansari. And I would like to thank extend my
Yes, sir.
thanks to Dr. Sonada for acceptance my invitation and giving us a good, excellent talk. Okay, thank you once again. Thank
I'm
you.
really thankful to you, sir, for inviting me to this. Thank you, sir.
Okay, over to Dhansari.
Yes, sir. Thank you once again, sir. And thank you all for joining. I hope it was a very insightful session and you enjoyed. And thank you and have a good day, all of you. Bye.